Zooming in into the liver histology, if we see these three entities, just put on the laser pointer. Oh, apologies. If we see these three entities, so a portal vein together with a bile duct and a hepatic artery, we're talking about the portal area where the blood enters the liver lobule and goes towards the central vein. Now here the lining of the epithelial cells, or the main epithelial cells, would be the hepatocytes, which are depicted here. And on top of them you have a very specific endothelial cell source, which is the liver sinusoidal endothelial cells, which actually have holes in them called fenestrae, which allows the blood actually going into the space of this and being in direct contact with the hepatocytes themselves. Now, hepatocytes is roughly 70% of the entire organ. We also have multiple other cell types here in a more schematic overview. So again, the portal area on this side, the central vein on this side. As I mentioned, 70% are hepatocytes, but we also have mesenchymal cell sources in the form of stellate cells. We have endothelial cell sources, as well as resident macrophages, being Kupffer cells, and bile duct cells. The liver is also zonated, so in the third region, more towards the central vein, we actually have phase 1 drug metabolism, and on the other hand, where you have more oxygen, so the blood is more richer, you actually have more beta oxidation, albumin production, all linked to the function of the liver itself. Now, why is it interesting to look at the liver in general in terms of in vitro modeling, but also ATMPs? We have a lot of different viral hepatitis infections, million each year, but we also have more fatty liver disease due to the fact that we all become a little bit larger, and 75% of the people that are obese have a fatty liver. Fatty liver is not a problem per se. 20% of these will progress into what's called a steatohepatitis with an inflammatory part, and also these, together with the hepatitis infections, will move towards end-stage liver disease where you have cirrhosis, and here only the curative treatment option for these patients is actually a liver transplantation. Now, also in the Netherlands, we see quite a lot of inherited metabolic liver diseases. 1 in 1,400 live births in the Netherlands have inherited metabolic liver disease. So think urea cycle defects or Crigler-Najjar, those types of diseases. Well, the only therapy for this end-stage liver disease is liver transplantation, and 22% of the people that are on the waiting list actually die before receiving a transplant. So we have a need for new curative treatments, and of course bioprinting could be one. Before I introduce the organoids themselves, let me take you back to the hierarchy of stem cells. On the left-hand side, we have the pluripotent cells, such as the embryonic stem cells, and really on the right-hand side, you have the unipotent cells, so terminally differentiated cells. Today, we're going to talk about adult stem cells, which are these multipotent progenitors. There are actually quite a number of examples of these adult stem cells, which is, for instance, the stem cells you have in the skin, but also the intestine. This is, of course, organs where we actually know that you have a lot of regeneration. So we know that there's adult stem cells within these tissues. And what's important to realize that these adult stem cells can only differentiate in the cell type of that particular organ. So an intestinal stem cell cannot create skin tissue and vice versa. These stem cells can be differentiated towards hepatocytes. But what we know from all the different sources of stem cells, that they maintain a certain degree of the function of a hepatocyte, but not all of them. So with stem cells, we say stem cell derived hepatocyte-like cells, because normally they have some of the function, but not all of the functions of the hepatocytes that we would like to see. This is a review that we published in Gastroenterology a little while back, indicating all the different sources of stem cells, including induced pluripotent stem cells, adult stem cells, and expandable hepatocytes, but also MSCs. And of course, our goal is to create these fully mature cells. Now we have a lot of strategies that we can actually apply with co-culturing, mimicking extracellular matrix, having a dynamic culture. This all contributes to the maturation of these stem cell derived hepatocytes. But now with biofabrication, we really can start combining all these different strategies and really mimicking the liver tissue in more detail and also with more function. So we're talking about the adult stem cells as Professor Clevers developed this back in 2005 together with Sato. They started with intestinal organoids, and here they isolated adult stem cells from the intestine, placed these in the laminin-rich hydrogels, and grew them out into these structures, cyst-like structures that he called organoids, because they mimicked the tissue in detail. After the establishment of the intestinal organoids, as you can see on the right-hand side, we already have a plethora of different organoid types that you can actually isolate and culture from different tissues, which includes, of course, also the liver. Just to give you an indication on what it actually looks like. So just envision you have this adult stem cell in this hydrogel, a laminin-rich hydrogel that continuously proliferates into, for the liver, a cyst-like structure. So as you can see, this organoid will start expanding to a cyst-like structure that's completely hollow on the inside. The way we propagate these cells is actually through a passage. You can do that by either adding an enzyme or doing a mechanical disruption with a pipette tip and then transferring it to fresh hydrogel. And this is exactly the way that we propagate these cells. On the right-hand side, you can see just after passaging, you have still these smaller fragments of these liver organoids, and you can see them growing out to these cyst-like structures again. And this happens in a matter of hours. So really the hallmark paper of the liver organoids, as they were called then, was this paper by Miri Hoog. What she showed is that from a very small biopsy, you can isolate the adult stem cells from the liver, place them in a 3D culture, and then expand them for months. The focus of this paper was really on the genetic stability of this particular cell type, due to the fact that they wanted to transplant these as a kind of an ATMP. Important to realize that organoids have two stages. We have an expansion phase, but also a differentiation phase. So in expanding conditions, you actually activate the Wnt pathway to a great degree, having loads and loads of cells. But if you take the most important growth factors out, they will start differentiating to hepatocyte-like cells. Because as with other stem cells, they will retain a form of the hepatocyte, but not completely. This is here also indicated with the albumin secretion. Albumin is one of the functions of hepatocytes. And if you have hepatocytes compared to the differentiation or DM condition, you can see it's 20-fold less. And also for one of the most important cytochrome P450s, the 3A4, you can also see it is over 20-fold less functional compared to freshly isolated hepatocytes. But what they did show here on the left-hand side, that they could transplant these organoids into immune-deficient mice, and just have to take it from me here. Here you see human hepatocytes derived from organoids that are actually engrafted in an immune-deficient mouse. So we're thinking about all the different sources. Today I'm going to talk mostly about liver organoids that are derived from the intrahepatic cholangiocytes, so from the smallest ramifications actually from the biliary tree, known as the canals of Herring. So these are the ICOs, as we call them. This is a bipotential adult stem cell, so it can differentiate in towards hepatocytes, but also cholangiocytes, and really depends on the media in combination with extracellular matrix composition. Good to know that we also have hepatocyte organoids, we have extrahepatic cholangiocyte organoids, and in terms of the liver itself, we also have gallbladder organoids. They all have different capabilities in terms of differentiation. Generally speaking, it's really the same culture method. And for the ICOs, as we call them, it's actually the most easiest cell source to obtain and also propagate. So in the first part, I wanted to discuss the in vitro aspects, because of course we want also to model liver function. And maybe the colleagues at REGENHU still recall this system. It was the 3D discovery that we used a couple of years back already with the bioprinter with the different cartridges. And in this case, we used a bioink very specific for the in vitro modeling. It was based on gelatin to create a liver construct. Now, why is it important to try to mimic? Of course, we need to induce the hepatocyte functionality of our models to test, for instance, new developed drugs. But since we also have organoids that we can derive from patients, we can also do personalized medicine approaches. And if you think about the gold standard, primary hepatocytes, there really is a problem because hepatocytes in culture de-differentiate quite rapidly. So if you see this, these are all hepatocyte markers. Even within hours to days, you see a decline of these hepatocyte markers. So it's really one of our goals to have liver function for a prolonged period that we can actually use to use as a screening platform for new drugs or existing drugs, but then patient specific. So in this scheme, we used the organoids and also organoids only. So we did not have supporting cells in this case, and we used the gelatin, which has methac-related groups, which means it's light sensitive. So if we have one layer of the gelatin, we come across with a blue light, it then solidifies, and then we build on top of that with new layers and new layers. Here you can see the printer in use, and really one of the main and biggest advantages is that we create a liver construct which is completely porous. So that means that our liver organoids are exposed to the media. They have these shear stress forces. So we combine it with a bioreactor, and there's an exchange of gases and nutrients in very close proximity to the cells. These are the bioprinted struts, and here with the H&E staining, you can see the organoids that actually reform within the bioprinted struts and are viable and functional. Since the liver is a very soft organ, we actually do pluronics, so a sacrificial material first to give initial rigidity. Then we come back with the gelatin with the organoids inside. Then it is covered with the blue light, it solidifies, and then we come back to the second layer and we build up until approximately 12 layers to have a construct of, let's say, 8 to 6 to 5 millimeters in size and roughly 1 million cells within the construct. The advantage here, again, we can do microfluidics. We can place this in a bioreactor and actually start testing under microfluidic conditions. Just to indicate that organoids, they are quite sturdy cells, but they don't like to be in the single cell format. So if you look at the live dead staining, basically all the organoids are very viable, but if you have single cells, they will die off. The viability of the printed organoids remains 100%. Here we measured this until 10 days, but we also have already data moving them towards 21 days. And again, here you can see the organoids in the bioprinted struts. So the bioprinting itself is very well managed by the organoids. Just to show you some gene expression results where we have a stemness gene that we measured in expanding conditions as well as differentiated conditions. But here we could also indicate what is differentiation in matrigel, plated in GELMA and printed in GELMA. So as you might expect, the stemness gene is completely gone when you have them in differentiating conditions. All these hepatic genes, they're more or less the same, but this one, the cytochrome P450-3A4, was induced actually with the printing itself compared to the plating in the gelma. So that was a really nice indication that actually the cells are more functional. And in this case, we did not even add the microfluidics yet. And we have already seen that you can induce this cytochrome P450 enzymes even further if you add microfluidics. So again, two applications that we envision with the in vitro modeling. We also have to increase the complexity to really induce more mimicking the liver tissue, let's say. So toxicology is one application that we do together with Dr. Kramer, and personalized medicine together with Dr. Fuchs. With metabolic liver diseases, we have vast biobank organoids, and there we can test what particular drug is actually good for the patient, which concentration, but also is it not toxic to the liver. Together with Dr. Kramer, we're looking more into toxicology effect of new or existing drugs to see if they are toxic to the liver, because many of those compounds that come into the market turn out to be toxic to the liver. So at the moment, just as an example, we're doing acute toxicity studies. So paracetamol overdose occurs quite frequently in the clinic. The functional part of that is acetaminophen or APAP. And if you look at the metabolic activity of the liver constructs, you can see in 72 hours that the cells will die off, also indicated by the live-death staining. The main advantage here, when we have those in bioreactors, we can also look at ATP leakage, but also very specific hepatocyte microRNAs, such as microRNA 122, to see if a compound is indeed toxic to the liver cells. So at the moment, we have tested, some of those compounds that we know that are extremely toxic to the liver, but also moderately toxic to the liver, or not toxic at all, really to validate the system. And then we combine this together with a bioreactor. This is a bioreactor that holds the construct within the bioreactor itself. It has a polycarbonate lid to observe the construct itself, but the bottom part is made from PEEK, so completely inert. It doesn't attach any of the compounds that we want to test. And here we can actually perfuse the construct itself with a setup, as you can see here. So in this case, we have a heat exchanger, we have the bioreactor and the pump all sitting within the culture stove. And this is a system where we can continuously perfuse, but also add the compounds of interest. And that's what we're doing now. So these are test compounds that we're now testing within the system. And you can see the construct here within the bioreactor while being perfused. Of course, this will be validated in the future to see if this can also hold true for other compounds, but the results so far are quite promising. When I'm moving towards the next step, thinking about using bioprinting also to create entire livers from scratch using the organoids, we have to envision that now you have these in vitro models with approximately 1 million cells. But if you're thinking about a human liver, we need more than 100, roughly 120 billion cells to create an entire liver. Of course, this is a huge leap, but we do have this really nice European project called ORGANTRANS. We're actually trying to achieve this. So this is the simplified overview where we actually have patient-specific cells. So we do an isolation of the patient. We have a mass scale expansion to create clinically viable number of cells. We then do a co-culture with supporting cells to really mimic the liver tissue. We create this as we call spheroids. I'll go to a little bit into more detail on how we do that in the project. We have a mass production, but also a quality control, very important. We have the bioprinting aspects and a maturation. This project will end with orthotopic, so in the liver transplantation, to show that these liver constructs can actually be maintained and are viable in mice. But the entire chain of events should actually be already suitable for human clinical application. Of course, also acknowledge all our partners, including REGENHU, that are helping with this project. So for the first part, the upscaling, this was actually quite successful by using spinning bioreactors. So our organoids within spinning bioreactors create vast numbers of cells, which means that with an inoculation of 2.5 million cells, we can actually achieve more than enough clinical viable number of cells in this particular technology. Then, of course, we have this vast number of cells that we want to combine with supporting cells. Why is this important? Well, of course, we want to mimic the liver tissue. We want to have an endothelial cell source to try to mimic this liver sinusoidal endothelial cells. But we also need the supporting cells that act like our stellate cells. And for that, we need a mesenchymal cell. So therefore, we use mesenchymal stem cells. And in this case, we use UVEX as our endothelial cell source. From our mass expansion of the organoids, we fragment into single cells and then mix in a 3:1 ratio in ultra-low attachment plates. These are microwells, basically inverted pyramids, where you have these cells in single cells. Due to gravity, they combine and create what we call spheroids. So in this case, we have tricultured spheroids with the organoids together with the endothelial cells and the mesenchymal stem cells. To show you here how that actually looks like under the microscope. So here we have the microwell plate on the right hand side. You have this mixture of cells. And you can see over time this runs only for 42 hours that the cells are actually interacting and creating these really nice multicellular spheroids. They form in roughly 4 days, but we harvest in between 4 to 6 days to take them towards the next step, which would be the quality control. This is one of the images of the microwells. So again, really nice formation of these spheroids. This is of course the best picture that I have. Usually you do see on occasion that you have spheroids that are larger. You have double spheroids within one microwell. And for this, together with one of our partners at CSEM, we developed also a quality control to be able to pick out what we call bad spheroids to make sure that we have a uniform spheroid in our mixture for the bio-ink, for the bioprinting itself. So this is the robotic sorter that we use in this case. So it's a system that can be placed in a laminar flute hood. Here you can see that there's a microscope. This is the well plate. We have a deep learning algorithm that indicates if you have good or bad spheroids. And here you can see one of the bad spheroids being picked out by this needle, being ejected from the plate. Thereafter, you actually flush out the entire plate and all the good spheroids will be collected in a 50 ml vacuum tube that we can actually use for our cartridge for the bioprinting itself. So just to show you a little bit on how these spheroids actually look like, you can see that there's an outer ring of the epithelial cells and the supporting cells are actually on the inside in this case. So if you do some of the stainings like the vimentin, which is one of the mesenchymal markers, you can see that they are on the inside. Keratin 19 epithelial marker, you can see nicely on the outside. What's also obvious from these transporters that we actually have the apical part of the hepatocytes on the outside and the basolateral part is actually on the inside of the tricultured spheroids. And these are the building blocks that we want to use in the case of the bioprinting. In the bioprinting itself, we also have to think about which hydrogel we want to use, but also we have to induce vasculature. So here in this scheme, we have the vasculature with the more opaque lines that we have lining throughout the constructs. And as a type of hydrogel of choice, we actually have a fully synthetic hydrogel. We cannot use the gelatin-based hydrogels anymore. These are animal derived, could be bovine, but also pig. So here we have a fully synthetic hydrogel based on PEG with the addition that is also hydrolytically cleavable, but also MMP degradable. So we know that our cells express a lot of matrix metalloproteases. So they will start eating away the hydrogel. And in the end, you'll have this full tissue graft without any of the supporting hydrogel present. And this is what we want to achieve within the maturation part after printing. So here you can see the construct already, and here the opaque lines, that's actually the vasculature that we induce. And I'll go a little bit into more detail on how we actually induce the vasculature. So this is the scheme as we have it. So we have within the printer the two VSDs. So this is extrusion-based bioprinting. On the left-hand side, we actually use cellulose, CNF. This gives the initial rigidity to the construct. Again, we have to print a fairly soft construct. The liver is a soft tissue, so we need this initial rigidity of the cellulose. And then as the vasculature, we have a gelatin-based hydrogel together with the endothelial cell sources. Here again, we use the UVEX. Then we have the OFD, which is recoined to the DFM, or the dynamic formulation module by REGENHU. And here you can see we have one of the packs. It's kind of a two-component glue that is mixed in a microfluidic chip. I'll tell you a little bit later on how we achieve this. And in one, we have the spheroids. We also have a media compound here and physiological salt for flushing. And here we can see that it's combined into a microfluidic chip. So again, here, just to show you where these spheroids then end up with, here we have the construct again, we have the vasculature running through this construct, and here you can see the spheroids nicely within the squares in between the vasculature. And of course, in the maturation stage, we want those to start to fuse and really create tissue next to a nice vasculature. So again, we want these spheroids to fuse within the construct, and this is really what we want to achieve within a couple of days after printing. And this project will end with the orthotopic transplantation. We'll first also do the kidney capsule to show the vascularization, where the recipient blood vasculature is actually combining with the vasculature that we have, and after that do an orthotopic transplantation in the mouse liver itself, in immune deficient mouse, of course, because we're using human cells in this case. So this is the bioprinter in working. So here we can see the bioprinter printing the construct. I think we just saw the CNF layers. And here you see the combination of the dynamic, the OFD, the print head with the cartridge. And here you can see the cells actually being placed into the construct. So with the setup itself, of course, everything is in a sterile environment, because we also need this completely sterile if we want to use it for transplantation. But there's also added features into this bioprinter itself. So it's more climate controlled. We have a fixed temperature within the hood itself to have everything standardized. And of course, we use this DFM multi-material dispenser, which you can actually tailor to your needs. You can also make different compositions of hydrogel. So there's a lot of optimization actually behind this part and this particular cartridge. Just zooming in a little bit more on the bioprinter itself. Here again, you can see we have a heated bed for the printing itself. We have the different cartridges. On the left-hand side, we actually use for the nanocellulose. The middle one is for the vasculature, and the epithelial cell sources are on the right, together with the spheroids. This is the chip that we're using. So in this case, we have a chip which combines the four sources of the DFM. We have one for purging, we have one for adding the cells, and then the two for the synthetic hydrogel, which basically combines and extruded outside of the needle into the construct. What we can also achieve here is that we can actually print straight into what's known as a substrate. So here you can see the construct being printed directly into the substrate, and this substrate can be placed into these 6 well plates, together with a smart lid, which is developed by one of the partners, CSEM. So here we can perfuse the constructs for multiple days to try to mature these building blocks to more functional tissue. And in terms of the vascularization, I can show you here that we have this vasculature with the GFP tagged UVEX that already shows that you have these endothelial cell sources. And this is what we're aiming to achieve. So to have these really hollow tubes with a patent lumen that really recreate the vasculature within the liver. Just to show you that this vasculature is also functional. So if you just add some dye on top of the construct, you can see that indeed this is perfused throughout the construct. So the vasculature in this case is functional as well. As you can see, the movement of this, the fluid, you can see the air bubbles throughout this construct. And this is really for us a new finding, but I did want to share you this results of the transplantation. So for the orthotopic transplantation, we're actually using a skin biopsy device, which is depicted here. So we make small holes in the liver of the mice, and in this case it would be immune deficient mice. We do the same for the construct, place it into the liver, and then add spongostan, which maintains the construct in place within the liver. And this is really, I don't think my colleagues at REGENHU already saw these results, but I did want to share this result with you, which it was, really just received this yesterday. On the right-hand side, we can see the liver of the immune deficient mouse, and this is actually the construct. And you can see here the spheroids within the construct, but also already creating this tissue-like mass within the construct. Now, of course, what we need to do here is use human-specific antibodies to really show that these are the human cells within the construct, within the liver of a mouse. But this is really promising results for us in terms of the efficiency of the success. Now, these constructs were maintained within the liver of immune deficient mice for 1 week, but we will follow up the mice up to 3 months to really show what is this tissue do, but also is it functional. So one thing that we will do, we'll take blood samples to see that there is an expression of human albumin within these immune deficient mice. And this together will show the feasibility of using these liver constructs as ATMPs for transplantation purposes. So maybe to recap a little bit on the advanced in vitro models as well as the whole organ engineering. If I think about advanced in vitro models, especially using these organoids, we can use them for personalized medicine. We have fast biobanks of our organoids that we can use and test particular drugs on particular patients, but also to see if those drugs are potentially toxic to that particular patient. We can test for drug-induced liver injury. So this is what we already validated within our system. This system that we have now does not have hepatic transportation. We really need other systems like transwell systems for that. So looking at drug disposition on the chip, we really need other sources. And what we also know already is that we don't really see hepatic zonation within our system. We get a kind of an intermediate phenotype within zone 1, zone 3. We do have the drug metabolism, which is very important for our systems. So the intermediate phenotype is not negative, but it's also not zonated within the structure. There are some micro-patterning papers out there that did show hepatic zonation with biofabrication technology. So whole organ engineering would really be the next step in liver bioengineering. And here it's really essential to look at the upscaling. We just need a huge amount of cells, but also being able to produce everything under GMP conditions. Autologous is an option, provided that the coagulation profile of the patient is good enough. We can take a biopsy from the liver, isolate the adult stem cells and grow them to organoids. Of course, for the HUVEX, we need another cell source. If we need this completely autologous, then we can do circulating endothelial progenitor cells. And for the MSCs, you can harvest them either from fat, but also from bone marrow. So is this really a truly alternate for organ donation? What we see now, at least in the mice, that it's feasible, but then still we have to make a really big jump towards human medicine. And of course, as a little bit of a commercial, next week we actually have one of our regenerative medicine workshop in Prague. There's still some places left here, so please register if you're interested in joining this symposium next week in Prague. With that, I would like to acknowledge my team and all the people that did the work. Of course, also all the different funding parties and REGENHU for providing me this opportunity to present this webinar in this series. So thank you very much for your attention, and I'm more than happy to answer some of your questions.
Thank you a lot, Bart, for the wonderful talk. It was a pleasure to hear about the advancement of this research and to see our DFM technology in action. You have been one of the first early adopters of it, and it's nice to show how great things can be done with that. So I invite anybody who might have question to use the Q&A section or the chat section of this meeting to post them. So I see we have already a couple. We start with, do you see potential for these organoids models within drug discovery?
Of course, our systems are more focused on high content screening. So for high throughput screening, I think there's definitely other systems that are more suited to high throughput screenings. So I'm thinking about the Mimeta system that is already using these liver organoids to do high throughput screening. So yes, in drug development, they could play an important role, provided that they have enough maturity. And what we see is that if you have the epithelial cells alone, you do get a certain degree of functionality, especially with phase 1 metabolism. But sometimes some of the cytochromes are actually lacking or very low expressed. So if we add supporting cells, we actually already see that we have more functionality in terms of this cytochrome P450 activity. And you may be also aware that Hans Clevers moved to Roche, and they're also really investing heavily in this organoid technology for drug development.
Another question, excellent work. A major challenge is to get enough cell to start with for producing organs from patient. How do you think this can be resolved?
Yep, of course, I would say that the spinner flasks, this is not a GMP condition, and we can create billions of cells in that regard, but we also need another system. So we're actually using bioreactors from Sinus Cell Expansion, which is a company here in the Netherlands that use culture bags, and we optimize the organoid also within this system. And here we really can create hundreds of billions of cells within controlled conditions. So what we actually see with more control, like oxygenator, pH, then we actually can achieve far greater numbers of these types of cells. So this is really the way to go. And of course, these systems will already have MDR, so they can be used also in a clinical setting.
Okay. We have another question more on the printing side. So the need for the sacrificial structure to mimic the vasculature. If you could repeat again what bioink was used to make the sacrificial structure and explain how this structure is then removed to.
Yep, I think I didn't indeed go into detail in this one. It's a gelatin-based material. We print this at 18 degrees, so then it's fairly thick. And after printing, the entire construct is placed under 37 degrees. So the gelatin will then become a liquid again and basically ooze out of the construct. Also, due to the fact that we have this in a bioreactor with microfluidics, the gelatin will be completely gone within a couple of hours, actually. But we print with the gelatin and the endothelial cell source inside. So they are on site. We don't infuse the endothelial cells. They are already there. The technology is actually based on a really nice publication, which is called void-free printing, where they showed that you can create this patent lumen with this particular technique.
Okay. Staying on the printing side, there is a question on the previous work that you showed, the one with pluronic as a sacrificial material, and they ask you about the UV curing process, how much you cure for each layer, which wavelength you use, and eventual effect on cell viability.
We did use UV before, but we use another photoinitiator, so currently are in the blue light spectrum. I think we used IGRA-Cure before as a photoinitiator. At the moment, it's blue light sensitive. Of course, you will have radical formation using this particular technique, but we've shown that the viability is from the organoids is really high, so they're not affected in any way with this technique.
Okay, and do you remind which was more or less the fabrication time for each one of these constructs? It was also part of the question.
Yeah, it's fairly comparable because they're also comparable in size. We don't need very large structures to transplant into a mouse. So they're 8 by 8 by 8 millimeters in size approximately for both conditions. And that means that we need roughly half an hour for printing one construct.
Okay. And now another couple of questions. So I see one on the differentiation. Do you think that the geometry of the constructs can affect their differentiation? So trying to create a similar geometry to the one that's found in the body could be the next step? Yeah, so we actually also showed this. That was with a volumetric bioprinting technique where we actually create what we call gyroids, so they have a more complex structure. And even when we corrected for the retention of the media in these constructs, we actually showed that certain morphologies actually increase the functionality. Of course, now with the resolution that we have now, we have perpendicular layers. And with the volumetric bioprinting, you can do, you have more flexibility in that regard. So that would be great if we could include that into the bioprinting constructs as well. But with the extrusion-based bioprinting, that's not feasible at the moment. And we have another regarding zonation. Do you think that directional perfusion can help establish the zonation?
Yeah, I think indeed this is the case. It's also shown in a paper that was on micropatterning with an oxygen gradient that showed that you can indeed mimic the zonation. For the ATMPs, I don't really know if it's an issue now, because we see that the tissue actually after transplantation will mature further into tissue. So I don't think that for the ATMPs it's an issue. It would be nice for the in vitro systems to really have this complete spectrum of hepatocyte function. I have to say, since the focus is on drug-induced liver injury, and that is mainly in zone 3, where you have the phase 1 metabolism. So as long as we have those cytochromes, we're actually already happy, but would be great to mimic. And microfluidics is one of the ways to go, for sure.
Well, thank you. Another last question, and then we can let you go. It's about mixing organoids with the hydrogel, and how do you do that, and if this caused eventual damage, or how you prevent causing any damage during the mixing phase?
So generally speaking, I would say that both the organoids as well as the spheroids are very sturdy cells. So they really can take a punch. What we typically do, we centrifuge into a vial and then add the bio-ink or the hydrogel that we want to print in on top. Basically have a pipette tip to take them up into the solution and then add into the cartridge. So yeah, the shear stress forces are minimal, and the extrusion-based bioprinting itself doesn't induce the shear stress forces. So they actually survive very well under these conditions.
Perfect. I will say that with this, we can close our session. I would like to take this time to thank you again for the wonderful talk and for being there and presenting us these activities. And wish you all and everybody who attended this event a nice day, and see you next time.
Thank you so much.